All right, we are back, and we are going to be talking about protein structure. So we talked about amino acids, what amino acids are, what they do, how they combine to form a peptide chain. Now we're going to see what happens when we form a protein. And so each protein, we're going to start with talking about protein structure. These are pretty well-known concepts that have been covered in a variety of different classes that you've probably taken, but we're just going to go over them again, reiterate some of the things, and then talk about how they're actually held together. So the first form of protein structure is known as primary structure. Right? Primary structure is just the sequence. Right? Is the amino acid sequence. We're going to show you later, not at the end of this video, but another video later, that primary structure is the only thing that's required for a protein to fold in its final three-dimensional structure. The second, right, so you have primary structure, and then we have secondary. Right, these are alpha helices and beta strands. Right, if you put more than one strand together, you get sheets. We'll talk about the directionality of these things in the next set of lectures. We're not going to really talk about secondary structure till later, and we'll get into a little bit more detail. The thing that stabilizes secondary structure is interactions between the backbone only. Right, so hydrogen bonding between the backbone, the nitrogens, and the carboxylic acid groups, or the carbonyl groups of the backbone, will stabilize secondary structure. What we're going to talk about and focus on right now is tertiary structure. Right, this is the final three-dimensional shape of a protein unit. It's what happens when you get long-range interactions between R groups after secondary structure has taken place. And then the last one is quaternary structure. Whoops, QUA. And this is multiple protein chains interacting. So things like hemoglobin would be an example of a protein that exhibits quaternary structure. So all proteins exhibit the top three: primary, secondary, tertiary. Only a choice few then exhibit quaternary structure if they're part of a larger complex. So we said tertiary structures are based on the long-range interactions between different R groups. So what are some of those interactions? How are they held together? We're going to look at some of the most common ones. The strongest one is a disulfide bond. Right, the reason why this is the strongest is because the covalent bond between two cysteine residues. So we have this portion of the chain. Right, so there's the backbone. Here's a cysteine residue. Normally it would have an H on it, but since we're going to form the disulfide bond, so the question is, when you form a disulfide bond, is that being reduced or oxidized? So we're going to oxidize these two sulfurs to form the disulfide bond, right? We're removing hydrogens. And so what is the best way to break a disulfide bond since it is a covalent bond? Well, let's use a reducing agent. So we can break it gently using a reducing agent, something like beta-mercaptoethanol. Mercaptoethanol. Right, so it looks like this. And what that will do is that will reduce that that disulfide bond. And what will happen is it will break it apart. And so let's redraw this disulfide bond a slightly different way so we can see it on a much larger scale. So here's our protein, disulfide bond. We add a little bit of beta Me, and what we're going to get is the reduction of our disulfide bond plus and the oxidation of the beta Me. And so that is the most gentle way to really break a disulfide bond. Another way we can break a disulfide bond is, let's go ahead and erase this. So that's for the reducing agent. Another way we could do it is we could oxidize it. Through an oxidizing agent, something like performic acid. So here's performic acid. We can add that into here, and instead of breaking it with H's, with hydrogen, since we're going to be oxidizing it, what we're going to add is this. And so what we're going to do is we're going to actually mark those sulfurs that were taking part in the disulfide bond. And in that case, we can further analyze to see which sulfurs are actually participating, right? We can separate these based on size because we've added now 3 oxygens to our sulfur, making them a little bit heavier. We've also highly charged them with those 2 negatively charged oxygens. And so we can separate these this protein in a specific way to see which sulfurs were taking part in the disulfide bond. So that's the strongest one. The strongest one is the disulfide bond. The next strongest, if you will, is known as a salt linkage. This is a quote-unquote biochemical ionic bond. It's the interaction between 2 charged R groups. So let's look at aspartate, which at pH of 7 looks like this. And then if we have lysine at pH of 7, it looks like this. Right, so when you put 2 charged groups together in close proximity, we form this salt bridge. So we talked about how to break a disulfide bond. We can use a reducing agent. We can also use an oxidizing agent. And those are the only 2 we can use because it's a covalent bond, unless we use extremely high heat where we're just really breaking everything. And so how do we break salt linkages? Well, we can change the pH. Right, so if we change the pH and we remove a proton off the lysine, or we add a protein to this carboxyl group, we can change, we lose that charge, and so then we stop that charge-charge interaction. This will actually be important when we talk about hemoglobin and the offloading of oxygen, because that's the technique that they use. The other way was we can use heat. Right, so just a gentle heating will break these 2 apart, as well as mastication or vortexing, or a physical force of some sort that can break it. And so the salt linkages are a little bit stronger than what we'll talk about next with hydrogen bonds, but they're still relatively weak in that we can easily break them through heat or through shear force. So then the next one is hydrogen bonding. Right, and so this occurs between hydroxyl groups of a serine, threonine, as well as a tyrosine. And so then we get, do this in different colors and actually let it show up. So we get these hydrogen bonds here. Hydrogen bonds, again, we can denature the protein. So if we break up any of these interactions, whether it be disulfide bonds, whether it be hydrogen bonds or salt linkages, we can denature the protein, will stop it from working, stop it from functioning. So there's a few ways with an extra way to break a hydrogen bond. And obviously it's the same way. Change in pH. Right? If we remove these protons, we'll no longer be able to form it. Heat, mastication, right? Because usually what we see with hydrogen bonding is their power is in the amount. One single hydrogen bond is not strong enough to hold two things together, but multiple hydrogen bonds, like Velcro, are. And then the last one, and this is why these things are so poisonous, is for heavy metals. Right? And heavy metals not only break hydrogen bonds, but they can also break disulfide. They can also prevent disulfide bonds from forming through a very similar pathway. And so let's say we have our protein, and so let's say we're looking at lead. The lead will actually bind to these oxygens, preventing, so it forms a covalent bond instead of the hydrogen bond. Hydrogen bonds, like we said, are strong bonds, quote unquote, but yet they still allow the protein to breathe, to move a little bit. When you start to covalently link those things together, you inhibit that breathing, causing more of a structure to that protein. And then the last form, the weakest types of interactions, are the London dispersion forces, the hydrophobic interactions, van der Waals interactions, however you want to call it. So the London dispersion forces is just between two nonpolar R groups, broken again by heat, agitation, or mastication. We're seeing a theme. However, we said this is the quote-unquote weakest interaction, and it is, based on what we've seen previously, but it's the one that actually kind of drives protein folding. So the majority of proteins are globular, right? They look like this big glob. And the reason why they look like this big glob is because of these nonpolar R groups. These nonpolar R groups want to get away from the polar water, and so what happens is they then enter into the center of this protein. When they do that, water is driven out, and it forces these polar R groups that can hydrogen bond, that can interact with the water, towards the outside. Right, where the water is. And so there's a lot about protein folding. We're going to talk about how we can purify proteins, how we can study proteins, and then we're going to get into more in-depth on protein structure and how these actually look, secondary structure, tertiary structures. And we'll actually see that if we can figure out how proteins fold, that would be really, really important, because right now we don't really know fully how they form or how they fold. We know the structures, and we know kind of about how they fold, and we're still learning more about making predictions on how they fold, but protein predictions is still a very inexact science.